Hello, welcome to another episode of Science Talks, a conversation hosted by the University of Arizona's Bio5 Institute. My name is Amy Randall Barber. The Bio5 Institute is home to hundreds of researchers and students, as well as many administrative staff who help support the cross-disciplinary research conducted here. Today we are joined by Dr. Frederic Zenhausern, who is currently the director of the Center of Applied NanoBioscience and Medicine at the University of Arizona College of Medicine, Phoenix, among many other appointments, including basic medical science, radiation oncology, biomedical engineering, and clinical translational science. Prior to coming to the U of A, Dr. Zenhausern co-founded and directed the Center for Applied NanoBioscience at the Biodesign Institute and the Flexible Display Center at the ASU MacroTechnology Works. He received his Bachelor of Science in Biochemistry from the University of Geneva, an MBA in Finance from Rutgers University, and his Doctorate of Science in Applied Physics from the Department of Condensed Physics Matters at the University of Geneva in Switzerland. Dr. Zenhausern is an inventor and a true master of interdisciplinary work and clinical translation. I am sure this only touches the tip of the iceberg of your achievements, founderships, and discoveries. Thank you so much for joining us today, Dr. Z. May I call you that?
Yes, of course.
So we'd like to get started with just a couple of fun icebreaker questions just to lighten the mood a little. So what was your dream job as a kid?
When I was a kid, I love animals and I wanted to be a vet.
Oh, okay. Awesome. Let's see. And second question, what would you like to be known for or remembered for?
Yeah, so I think what is interesting is my training and also through my professional career. I am what I would call a true interdisciplinary scientist. And I think I demonstrated how, in fact, interdisciplinary science can bring, in fact, ideas across many different areas of scientific and engineering technologies, really ranging from discoveries. So I developed some basic optical sciences when I was at IBM, to product, in fact, platform when I was at Motorola Labs, or even now at U of A, where we develop technology that goes through the FDA and ultimately reach the marketplace. And really bring also practical solution in life sciences, biodefense, and healthcare. So we have a very broad portfolio of technology impact.
Yeah, wow. Yeah, I was noticing that when I was reading about you prior to coming. But I mean, like this says here, master of interdisciplinary work and clinical translation. So that's just exactly what Bio5 is all about. So Thank you for being a member here and contributing.
Thank you.
So you started off in Switzerland and you ended up in Arizona. Can you tell us how you ended up in Arizona?
Wow, that's a long journey and a long story. So what happened is when I was in Switzerland, I did my PhD thesis in partnership between the University of Geneva, University of Lausanne, but also IBM research in Zurich. And of course, after my PhD, I was looking for a postdoc position, and I got a beautiful location at UC Santa Barbara with a nice beach, and my wife was very happy that we were ready to move to California. But IBM really twist my arm and say, you should come and work for us at the IBM Research Lab, which is located in the Bronx. So I say it will be a difficult sell, but no, ultimately it was a great experience. And again, going to New York State was amazing. So I stayed many years at IBM Watson Lab, where I developed again different technology platform. From DNA sequencing, I was maybe the first one also to bring a real virus at IBM to look at using our high-resolution microscopy techniques to image those particles. And we did a lot of different discoveries as well. And then that really brought me then to develop all kind of technology. And I moved, in fact, to Princeton, New Jersey, where I was part of a Swiss chemical company, Firmenich, where I also developed new technology for mimicking the nose, the human nose, looking at electronic nose technology. So I was really working in Princeton, New Jersey with a chemical company, but also I joined, in fact, the Princeton Optophotonic Center as an industry member, and really developed some research there, which ultimately led me also to decide to start a new startup company in the Princeton, New Jersey area, which was Alphamos America, which was again a subsidiary, in fact, from a French startup as well, that ultimately we brought to the stock market to commercialize that electronic nose technology. Which was also interesting because at the time we were also discussing with David Walt at Tufts University, who had also an interesting technology that ultimately was used to start Illumina, that we all know now in the field of DNA sequencing. And David was a good friend, and ultimately, interestingly enough, they never used his patent related to electronic nose technology. So that's the small anecdote here. But ultimately, I got also recruited by, in fact, one of the advisory board members of the company that was also part of Motorola. And they were establishing a new biosystem division here in Arizona to start the DNA microarray technology. And I got recruited, in fact, to come to Arizona. So at the time, we were still debating, they were still debating if it will be in Chicago or Arizona. So we got lucky, we ended up in Arizona.
Yeah, definitely. The weather is much better here, I think. There's a lot to do in Chicago, and they've got
That's
the lake, and you know that part is nice. But
and the cold.
Yeah, but it's cold. Yes, absolutely. Okay, well, yes, very long journey to get here, but I'm glad you're here. Thank you for accepting. So you were inducted into the National Academy of Inventors as a fellow in 2013 for the invention of a rapid DNA processor. You mentioned this just a moment ago, working with the inventor Illumina, isn't that? Yeah. And so can you just kind of explain a little bit more about that and how we use it today, or how it's evolved since then?
Yeah. So it was interesting because also the goal for us has always been to try to do molecular diagnostic. And when we started the technology using mostly microfluidic devices to, in fact, automate and simplify the workflow process for preparing a specimen, we realized that any application in medicine will be a long project for us because we need to go through the FDA for regulatory compliance. And maybe we're not super ready for that at the time. So we look also at other applications where maybe there were some element of regulation, but not as stringent, like going to the FDA. So that was a time where really there was a big backlog of DNA fingerprinting. It was taking years until a sample could be processed, and ultimately the Department of Justice decided to promote, in fact, new technology, trying to solve that backlog. So we came, in fact, with a technology that was trying to reduce all that workflow process from the laboratory and bring it into a small machine. And that's what we did initially with the contract with the FBI. And the FBI introduced us also to other countries and other police forces, including the UK Forensic Science Service, or FSS. And they were, in fact, really the inventor of the technology with some group at the university in the UK. They were the world leader in that technology, and they really had the vision of, can we bring that technology closer to the crime scene? So initially we developed a technology that was supposed to go into a van that goes with the police. But that scenario changed, and ultimately the technology has been deployed at the police station. But that allows, in fact, to screen, potential offenders sooner, and potentially, of course, crime sample, much easier. Again, there was a complex regulatory validation. So we were, in fact, a part of a large European program as well, where we validated the technology between the police forces in the UK, Germany, Austria, and the Netherlands. And that was a large European program called MIDAS Consortium. And ultimately, we got successful with that project. Coming back then to the US, ultimately the FBI continued the work, and to the point that in 2017, they modified the DNA Act by Congress to include Rapid DNA Act. And that was signed also by the president in 2017 to approve the use of those technology now in court. And they started to get some commercial development, and the technology now is used ultimately in the Justice Department.
Wow, that is, I'm just blown away, you know, and as you mentioned earlier, what you do applies in so many areas, and that's, wow. You are currently the director of the Center for Applied NanoBioscience here at U of A. Can you briefly tell us about the research that goes on there?
So we have a large portfolio of activities right now. So our center is about a group of about 15 members. We are very interdisciplinary scientists or researchers. We have mechanical engineer, physicist, MD, PhD, molecular biologist. So we can really do a lot of different projects, because our goal is really to identify, in fact, medical needs or problem in the healthcare delivery, and try, in fact, to find solution by, in fact, applying some engineering principle to try to come to those solutions. And we love also to bring teams, because we think that solution from the discovery to the implementation require a lot of different people. So we like to work with different agencies, but also, of course, industry partners as well. So right now, our portfolio is very broad. We have, program with NASA, NIH, DoD, and typically we have very early stage discovery project. So, for example, we look at new drug delivery system, as we all experience with the COVID-19, revolution of the mRNA delivery using those nanolipid particles. Now what we are doing is trying, in fact, to use, in fact, plant-based lipids that, in fact, the plants are secreting as a potential drug delivery system, because plants have an intrinsic agent. For example, they can be anti-inflammatory, antibiotics, or antioxidant. And we can also load those particles now with those different drugs, or we can also do gene delivery using those plants. So still relatively early stage, technology platform to, in fact, large scale, manufacturing capabilities that we have. So we have developed also a platform technology, an in vitro system that potentially could replace also the animal model for testing drugs. Our system is called an organ-on-chip platform, where we combine also the organ-on-chip and organoids, which is that 3D cell culture capabilities. And right now we have been scaling up that technology to the fact that we have a partnership with Mitsubishi Gas Chemical Company in Japan that is now mass manufacturing our devices, and we will also embark into the commercialization of that technology that ultimately will be useful for testing drugs and personalized, in fact, treatment for the patient. So either, for example, at the hospital, if a cancer patient, we can extract some of the cells from the tumor, treat that tumor into that model, and look at different combination of therapies that might be more appropriate for the person, while you're also profiling the genomic signature of the tumor as well. So you can really adapt those therapy. And one area where we are also expanding is radiotherapy. You can also personalize radiotherapy much better by those techniques. So we have a very broad spectrum of activities.
That's awesome. So just how long before something like that is actually, how long does that take? 10 years?
So it depends. So for example, just to give you some ideas. So for example, when we talk about the rapid DNA testing, it took about 15 years to get the technology from the development to the implementation. A COVID test that we did that is approved by FDA that you can buy on Amazon now has been developed in less than a year.
Yeah.
And now those kind of platform technology for organ on chip will take a few years, about 5 years until they can be really deployed in the marketplace. But it will be still a research platform at that level.
Yeah. Okay. Well, thank you. Just curious because that is just amazing technology, and it will be so needed. It is needed, and
But that's also, it's a very good point. Is sometimes we think about also getting a grant. It's only 5 years, and it's most of the time not enough to mature technology. So what are the mechanisms in academia to allow us to keep going and be part of that development, because there is still a bridge until really a company can take it. I think it's interesting. There are few mechanisms that the government is offering, but not many. And that's why I think, getting to the university vision and sustainability of developing those kind of technology is very important.
Absolutely. Can you tell us what your inspiration was for getting into this kind of work? Did you always want to do this kind of interdisciplinary work, or did you just sort of end up here?
It's a very good question. So as I said, by my training when I was in college, I was in biochemistry first. And when you reach your senior year, you need to do a research project. And that was just at the time when, in fact, IBM Research in Zurich got two of their scientists got the Nobel Prize in physics for the discovery of what we call the scanning tunneling microscope. And that was a microscope that can profile surfaces and see atoms. And I was fascinated by that technology, and I thought that, it could be useful to look at molecules as well. So I contacted, in fact, those scientists at IBM, and I said, I would like to come for my summer internship in your lab. And Ainiro, one of the scientists, said, I don't think we have anything for you. We don't do biology. We do physics. And he said, but okay, let me think about it and call me back. Call him back, and ultimately I got the internship, and we didn't know what to do. But ultimately we started to look at molecules using that technology. So I got so excited by the experience that ultimately when I came back, I decided to enroll in a PhD program, and I decided to switch to physics. That's how I got excited by that, and everything else then went.
Yeah, okay. Yeah, and sometimes it just takes to be subjected to something or to change your mind.
Exactly.
So you've done a lot of work in very scientific focused arenas. However, you achieved your MBA in finance. Do you think that has helped you in your career?
Yeah, so first of all, we have to put in context when I did that. So it was also in year 2000. I was living in the New York area, and the economy was booming. Everybody was thinking that, if you go on Wall Street, you can start a company, you can be rich. And they were really tapping into a lot of scientific talent that they realized could be doing a great job on Wall Street. So in fact, a lot of mathematicians and physicists from Princeton ended up in the venture capitalist group and stuff like that. So I got also, I think, under that influence a little bit of that time. But I also realized that one day I wanted really to do a startup company, and having the financial skills and managerial skills will be really helpful. So that's why I enrolled in the program at Rutgers and ultimately got my MBA. And I focus on finance because, again, of that environment. And I think finance was a little bit easier for scientists because there's a lot more math and thing than maybe the other soft skills, topic. So, but very useful. I think I learned a lot, in fact, with those soft skill classes as well. There are topics that I was maybe not trained into, looking at them and they are very important when you manage people or interact with people. So ultimately it was very useful, and I think I apply some of those principles all day around.
Yeah, okay. So what are you currently looking forward to in your academic or personal life? It could be anything. It doesn't have to necessarily be scientific.
But I think, at the personal level, I think I am very privileged. I have two outstanding children, adult children now, who are pursuing exciting, venture in medicine and engineering. So
wow.
I'm very lucky with that. While in fact also family is growing and getting grandchildren, so I'm very happy with that.
Yep.
But I would be also interested, in my academic, or professional life to really keep going and serving the community, trying to look at some of those grand challenges in society, and still looking at those programs in terms of our work to try to address some of them. And I think also because the economy is changing. Right now we are really at the intersect of also climate tech, biotech, and space tech. We are exploring and going to the universe, and I think it's very exciting to see those kind of synergy of technology as well. So I think we have a very exciting time in front of us, and I hope that I will still be able to contribute to that world as well. So. And I think that's about it. I think, I love mountain biking too. So maybe I would like to start a new UA, Phoenix club, so we can go riding as a group.
That would be cool. That would be cool. There's another researcher that I just talked to in Tucson, though, but that also mountain bikes all the time and lives out by Star Pass and just goes through that area. And,
that's nice.
Yeah. I think you should do it.
No.
You should do it. Well, we thank you again for being here with us today and telling us all about your journey and your inventions and everything you're doing now as well. So thank you so much once again.
Thank you very much, and we're happy to be Bio5 members.
Thanks to our listeners for tuning in to another episode of Science Talks. Continue the conversation with us next time as we learn more about the amazing science happening at the University of Arizona's Bio5 Institute.